All right, well, good afternoon and welcome back to Science in the Age of COVID-19. Today we are super excited to have Jesse Bloom, a Hughes investigator associated with the Fred Hutch in Seattle. The Bloom lab studies viral evolution. Historically, they've done mostly influenza and HIV, but they got the memo loud and clear, like many other researchers did, that it was time to retool the research program to take on COVID. The approach that he's going to tell us about today, several approaches, but a big one is that they make and experimentally characterize all possible mutations on pathogen proteins to see what sort of evolutionary space is available to them and how a host might adapt. And so he's trying to predict the future of what sorts of nefarious activities this pathogen will be able to get up to. In addition to doing fantastic science, Jesse is deeply committed to open source. So they make a lot of computational tools, which they make freely available. All his data sets. If you're here from Twitter, you will notice that he provided his slides, and we will post a link to that, or maybe it'll be on one of his slides. So we're super excited. So Jesse, take it away.
Thank you. Hopefully people can hear me. If you can't hear me, someone somehow let me know. So I'm Jesse. I'm going to talk today about the evolutionary potential of the SARS-CoV-2 receptor binding domain. As was just mentioned, I'm at the Fred Hutch Cancer Research Center and also an investigator with HHMI. And the slides that I'm presenting today are available at this link in yellow on this slide here. So you're welcome to go to that link if you want to follow along or follow any of the web links that I'm going to present. So SARS-CoV-2 virions are decorated by spike proteins, which are shown here in red, that mediate viral entry into target cells. And one important part of the spike is the receptor binding domain, which is shown here in purple. So this is an autonomously folding subdomain of spike. It's about 200 residues long, and the spike itself is a trimer of proteins that are about 1,300 residues long. And the RBD is normally sort of packed down against the rest of the spike, as shown here, but it can transition to sort of this up conformation where it's pointing further up from the spike. And when the RBD is in this up conformation, it can then bind to ACE2, which is a protein expressed on the surface of target cells. And so the binding of the RBD to ACE2, as sort of illustrated here, is the first step in viral infection. And so this is sort of a necessary step in order for a virus to be able to infect a cell. In addition, we now know that neutralizing antibodies to SARS-CoV-2, not always, but quite often, probably the majority of the time, bind to the RBD. And in many cases, they actually act by simply blocking the interaction of the RBD with this ACE2 receptor. And so for these reasons, really understanding how mutations affect the RBD is crucial for understanding the evolution of SARS-CoV-2. And it's crucial in sort of two contexts. First of all, for understanding the deeper sort of origins of this virus and evolution of the larger family of SARS-like viruses. This virus's ability to infect humans depended on, at some point, this virus acquiring the ability for its RBD to bind well to the ACE2 on human cells. So that's sort of a deeper evolutionary question, which I'm not going to really get into today. It's also crucial for sort of understanding the ongoing evolution of this virus and things like antibody therapeutics and vaccines right now, because as I just described, most vaccines and antibody therapeutics rely on antibodies that are targeting the RBD and trying to block its interaction with ACE2. So we're all very accustomed in biology to the idea of mapping the sequence of a protein to its structure. This is basically what's known as the field of structural biology, and this is often done, for instance, by using X-ray crystallography or cryo-EM. I'm going to be talking today about a different kind of mapping, which is not the mapping of sequence to structure, but is rather the mapping of changes in sequence, so mutations, to the biochemical phenotypes that we think are relevant for the function of a protein. In this case, the protein being the SARS-CoV-2 receptor binding domain. And in the case of this protein, we think that three highly relevant biochemical phenotypes are: the protein has to fold properly, it has to bind to ACE2, so that's sort of its function. And then with respect to sort of our interest from public health, whether or not this protein can be bound by antibodies is highly relevant to the effectiveness of these therapeutics. So everything I'm going to be discussing today is experiments that map from change in sequence to these biochemical phenotypes, but we're still going to talk a lot about the structure of the protein, because even though we're not measuring any structural properties here, structure still matters because it's the structure of a protein that implicitly determines the majority of this mapping from change in sequence to change in biochemical phenotype. So my talk is going to break down into four pieces. First, I'm simply going to talk about how, from an experimental perspective, we map how RBD mutations affect these biochemical phenotypes of folding and ACE2 binding. And I'm going to show you what these maps look like. I'm then going to discuss how we can also map how mutations to the RBD affect the ability of antiviral antibodies to bind to this viral protein. And then I'm going to transition to talking about how we can use these maps of the phenotypic effects of mutations, in particular, how we can apply them to try to predict if or how the virus might evolve to escape from antibodies. And then finally, I'm going to touch briefly on how we can use the maps to inform surveillance of ongoing viral evolution in nature. So first part first, I'm just going to discuss how we make these phenotypic maps of how RBD mutations affect folding and ACE2 binding. So there are a lot of possible mutations you could make to this receptor binding domain. It's 201 residues long. Each residue can be mutated to 19 other things. So that's 3,819 possible amino acid mutations. So in order to look at the biochemical effects of these mutations in high throughput, we used a yeast display system. So as I mentioned, the RBD is an autonomously folding protein subdomain. So we simply express it on the surface of yeast. And in general, only proteins that are properly folded will be trafficked to the surface of yeast. And then we can detect them with a fluorescent tag on the RBD. We can also incubate these yeast with fluorescently labeled ACE2. And to the extent that the RBD binds to the ACE2, the yeast will then become fluorescent with that particular color. Importantly, we don't just have sort of yeast expressing one RBD sequence. We actually have a library of yeast, each of which is expressing a different mutant of the RBD. And these yeast are also carrying 16 nucleotide barcodes that tell you which RBD mutant they have on their surface. And I'm not going to go in details right now about how exactly we link up this barcode to which RBD mutant the yeast has, but you can click here if you want to read details on how that's done. So if we didn't have this library, if we just had a single yeast variant, it's easy to see how you could actually combine these yeasts with flow cytometry to essentially make biochemical measurements. So let's say you just had yeast that were all expressing the same RBD variant. You could take these yeast and you could incubate them with fluorescently labeled ACE2 at different protein concentrations. And then if you ran flow cytometry on them, this is what you'd expect to see. When you have very little fluorescently labeled ACE2, such as at the top, there's going to be no fluorescent signal from the ACE2 because there's nothing to bind to the yeast. And as you increase the concentration of the fluorescently labeled ACE2, it's going to start binding to the RBD on the yeast, and then eventually at high ACE2 concentrations, you'll saturate that signal. And so if you were to just plot this fluorescent signal as a function of the ACE2 concentration, so essentially flip this plot on its side, you would get what we call a biochemical binding curve, where you can see the binding fraction, or the amount bound, is a function of ACE2 concentration. And you can use that to determine things such as the dissociation constant for the binding of the RBD to ACE2. Now, as I was describing, we don't just have a single RBD variant. We have many, many thousands of different RBD mutants. but we can actually sort of do the same experiment to measure binding by all of these mutants all in one experiment. And so what I'm going to describe here is a deep mutational scanning approach, which is an elaboration of a strategy called TightSeq. So essentially what we do is we take this whole library of yeast and we label it with different ACE2 concentrations, just as I was describing before. And then just as if there was one RBD variant, sort of any RBD variant is going to start to move to the left in this flow cytometry, in these flow cytometry plots, as we increase the concentration of ACE2. But different RBD mutants are going to move over sort of at different rates. So rather than reading out the fluorescent signal, we instead sort the yeast into 4 bins and then just sequence the barcodes to tell us which RBD mutants are in which bin at what concentration. And so doing that, we can build dissociation curves, just like I described to you, for all of the RBD mutants sort of in one experiment. And then we sort of do something similar where we just look at the fluorescent signal from the tag on the RBD in the absence of ACE2 to measure expression of folded protein for each mutant. So the result of these experiments is sort of a lot of data about the biochemical phenotypes of these mutations. So I'm going to show you a little bit about what these data look like. So if you click on this link here, it should take you to this heat map here. And so this heat map is now showing these 2 biochemical phenotypes that we think are related to the function of the RBD. At the bottom is their properly folded RBD expressed on the surface of the yeast, and at the top, how well does it bind to ACE2. And then each cell here is a different mutation, and they're colored such that mutations that are bad for these biochemical phenotypes are red, and mutations that enhance the biochemical phenotypes are blue. So for instance, you can pick any mutation. So let's say mutation V350D, and we can see that that mutation is very bad for ACE2 binding. So it's very red there, and it's fairly bad for expression of properly folded RBD. So it's moderately red at the bottom. And we can sort of scroll through these maps and just look at the effects of mutations at lots of different sites. And you'll see there are some regions of the RBD that say like site 431 that are highly constrained with respect to ACE2 binding or folding. There are other sites that say here like site 441, which is pretty tolerant of mutations. Most of them don't have much of an effect. And you can even find scattered mutations such as this one here that enhance these properties. So Y453F actually enables the SARS-CoV-2 RBD to bind more tightly to human ACE2. And you can also identify mutations such as L455K, which increase the expression of properly folded protein. And in general, these mutations actually enhance the stability of the RBD. So this is sort of a lot of data. And if you're not sort of deep in the details of the RBD, it looks a little bit boring and overwhelming, but we actually think these data are quite useful for people who are doing things like engineering the RBD for incorporation in vaccine immunogens or looking at the natural evolution of the virus, and they see some mutation and they want to go look up and see what it does. So it's sort of a lot of data, but it's sort of a useful reference to have, sort of like a genome or something like that. Okay, let me go back to my I get back to my slides. Okay, so we can also project these phenotypic maps onto the structure of the RBD. So as I was describing to you, the effects of mutations are largely determined by the structure of the protein. So here is the RBD shown here in sort of this light gray or white color. And here's the ACE2 that the RBD binds to that is shown here in sort of this darker gray. So for instance, right now I'm plotting, here's the primary sequence of the protein, and I'm plotting the average effect of mutations on the ability of the RBD to bind to ACE2. So for instance, if we look at mutations that are bad for the ability for RBD to bind to ACE2, most of them occur on this region of the RBD that's actually in contact with ACE2, which sort of makes sense because if you disrupt the contact interface, the binding will get worse. We can also look at where there are mutations that affect the sort of folding and expression of this protein. And those mutations, if you look at the sites where those mutations are really bad, you can see that they tend to sort of be in the core of the RBD here, which is what we'd expect, because proteins often derive a lot of their stability from sort of the core packing. So I think this just sort of illustrates how we can interpret these biochemical phenotypes in the context of the protein structure. Okay, so I hope I've told you how we can just sort of build these big phenotypic maps. And before I really tell you what they're useful for, I want to tell you how we can first layer on one additional phenotype that's of a lot of public health relevance right now. So I'm going to show you about sort of generating even more data, and then we'll get to how we can use those data. So I'm now specifically going to describe how we can map how RBD mutations affect the ability of antiviral antibodies to bind to the RBD. So as I mentioned at the outset of the talk, most characterized neutralizing antibodies to SARS-CoV-2 bind to the receptor binding domain. Not all of them. There are some that bind to other regions of the S1 domain of the spike, but the majority bind to the RBD. And right now I'm going to describe the following antibodies. 9 that were isolated from SARS-CoV-2 patients from our collaborators in the Crow lab at Vanderbilt, and then 1 cross-reactive antibody that was actually isolated from a SARS-CoV-1 patient, but that binds to SARS-CoV-2. And so in order to map the mutations to the RBD that can escape from binding to these antibodies, we do another really simple experiment. We just take this library of yeast, we incubate them with a fluorescently labeled antibody, and then we essentially sort for the yeast cells that are binding to less antibody than most of the population, after doing some filtering to remove those that just fail to express folded RBD at all. We then deep sequence the original population of yeast and the yeast that have RBD mutants that can escape antibody binding, and then we look to see which mutations are enriched in the antibody escaped population. So in this cartoon, the escaped mutations tend to be things like mutating site 483 to a threonine or 484 to a serine. So I'm going to draw logo plots where I'm going to put really tall letters where there are mutations that escape binding by the antibody. So what are these? So as I said, tall letters indicate strong escape mutations. So what do these data actually look like in real life? So here's the escape map for just one antibody, CoV2 2499, and I'm just showing a subset of sites in the RBD that are sort of relevance, including all of the sites where mutations have a big effect on enabling the RBD to escape from binding by this antibody. And so you can see that these viral escape mutations are clustered in 2 regions of primary sequence around 440 and around 490. And again, we can interpret a lot of the phenotypic effects of these mutations in the context of the structure. So if we go back to this sort of interactive view here, okay, here's the RBD in this sort of light gray color, and here's ACE2 in this dark gray color. And here are the primary sequence of the RBD, and the point shows how much mutations at each site escape from the antibody. So for instance, site 447 is the site of the strongest escape mutations. And so if we look and we see where those escape mutations actually fall in the structure, you can see that they all fall in this one localized region on the RBD, which must be where the antibody binds. And so this sort of makes sense that the escape mutations are generally in the region where the antibody binds, although it turns out that not every residue that contacts the antibodies tends to be escape mutations. There's a lot of sort of hot spots of escape mutations. Okay, so that's what one of these escape maps look like. And the cool thing is we can do these experiments for lots of antibodies quite quickly. So before I show you more of these escape maps, I'm going to try to make it easier to mentally orient what I'm showing. So I'm going to divide the SARS-CoV-2 receptor binding domain into 3 sort of classifications. So the first classification is just the structural core of the RBD, which is, I'm going to illustrate by orange. So none of these sites are orange because this antibody doesn't bind in the core of the RBD. Then the receptor binding motif, so residues that are in the part of the RBD that contact the ACE2 receptor, I'm going to color in light blue. And then the residues in the RBD that actually make direct physical contacts with ACE2, I'm going to color in dark blue. So you can see this particular antibody is escaped by mutations that mostly occur in the ACE2 contact residues within the receptor binding motif. Here's just escape from a couple of additional human antibodies to SARS-CoV-2. And I hope what you can just appreciate by looking at these is that all of these antibodies are primarily selecting escape mutations in these ACE2 contact residues. So in a sense, all of the antibodies bind to a generally similar region of the RBD, but yet they actually have quite different escape mutations. So for instance, this antibody at the bottom and this antibody in the middle have no escape mutations in common. And this antibody at the top and this antibody in the middle share escape mutations at some sites, for instance, 487, but at other sites like 486, mutations only escape the middle antibody and not the top antibody. And you can see the top antibody can be escaped by a mutation in one particular region of the core of the RBD where the other antibodies can't. And we've done a lot of viral neutralization assays to actually sort of confirm that these mutations that we're mapping here do indeed validate in terms of the effect on actual viral neutralization. So as we generate more and more of these escape maps, it becomes useful to sort of be able to organize the data. So here I'm showing a multidimensional scaling plot that allows us to organize antibodies in the space of viral escape mutations. And so for instance, if you're used to single-cell RNA-seq, you can sort of think of this as conceptually similar to a TS&E plot. It's actually built by multidimensional scaling, so it's a different algorithm that creates the plot, but the idea is the same. We've projected this high-dimensional information about viral escape mutations into a two-dimensional space where antibodies with similar escape mutations are close to each other. And furthermore, each antibody is illustrated by a pie chart, which tells you which regions of the receptor binding domain it selects escape mutations in. So for instance, antibodies 2165 and 2832, if you look over here at the details on the right, share some common escape mutations, such as 487. So they're close together in this space of escape because the same mutation can get away from both of them. Antibody 2499 at the bottom here has no common escape mutations from these other two, so it's located a long ways away in the space of viral escape mutations. And we now have, we haven't quite finished polishing the data, but we now have these sorts of escape data for dozens of additional antibodies, including some that are actually being advanced in human therapeutic trials. And we think it's really nice to be able to take this sort of rich escape information for dozens of antibodies and sort of project it down into this two-dimensional space to really understand which antibodies are similar and different in terms of the potential for viral escape mutations. Okay, so I've now told you two very dense sort of data generation vignettes. So particularly how we can map how mutations to the RBD affect its function, so its folding and ACE2 binding, and then how mutations affect its ability to be recognized by antiviral antibodies. And so now I want to tell you about how we can actually use these maps to sort of understand viral evolution under antibody pressure. And I think this is really cool because it illustrates how understanding of sort of the microscopic effects or the biochemical effects of mutations can actually inform understanding of these higher level evolutionary processes. So we do think that there's potential for SARS-CoV-2 to eventually evolve in response to antibody pressure. It's not clear how much that will happen. But in order to sort of let us simulate this evolutionary process in the lab, we grew a spike-expressing virus. So it's actually not SARS-CoV-2, it's VSV. And these papers have a lot of details about that. I'm not really going to go into it in detail, in the presence of antibody. And this allows us to see what viral mutations are actually selected by the antibody. And again, these links have more details if you want to read about exactly how the assays work. So for instance, if we grow this virus in the presence of this antibody CoV2 2499, we did that 16 different times. In 5 of these 16 times, we selected viral escape mutations. The other 11 times, the antibody was sort of able to just keep the virus under control for the duration of the experiment. And those escape mutations were G446D 3 times and Q498R 2 times. So now I want to ask, can we go back to our biochemical maps of the phenotypes of mutations and understand sort of why the evolutionary process went the way it did here? So first, I'm just going to show you our map of which mutations abrogate binding of the antibody to the virus. And here are all the mutations that eliminate binding of the antibody to the virus. So you can see G446D right here and Q498R right here are indeed two of the mutations that abrogate antibody binding. But there's a lot of other mutations, so it's not obvious why these were the two that actually arose. So one thing we can do is we can notice that in our experiments, we were looking at all amino acid mutations, but the virus actually evolves by single nucleotide changes. So now I've subsetted the map of amino acid mutations that escape antibody binding down to just those that are accessible by single nucleotide changes. And you can now see that there are fewer possible escape mutations that are evolutionarily accessible, and the two that are actually observed are retained in that set. Now, importantly, this map just shows which mutations escape antibody binding. But we think the virus also needs to retain its function, such as being able to bind to ACE2. So as I described at the beginning of the talk, we've also measured how all mutations affect ACE2 binding. So now I'm coloring these mutations by how well the virus with that mutation is still able to bind to ACE2. And so mutations that are dark red or brown retain good ACE2 binding, and those that are faded to yellow bind to ACE2 very poorly. So you can now see that there are only a handful of mutations that are evolutionarily accessible, retain good function, and escape antibody binding. And the ones that actually arise in the virus are among this small subset of mutations. So another way to present those same information is shown here. So now I'm showing a scatter plot for this particular antibody CoV2 2499, where I'm showing two different biochemical phenotypes on the two axes. So the x-axis is how well that mutation, that viral mutation, escapes antibody binding. And the y-axis is how well that mutation retains binding to ACE2. So if you look at this plot, you would expect the virus could actually evolve to escape from antibodies by mutations that are sort of in the upper right here. So the mutation needs to escape the antibody, but it still needs to retain good ACE2 binding function. And in particular, when I gray out with these gray X's, all of the mutations that are not accessible to the virus by a single nucleotide change, you can see that there are actually only 4 mutations that are strong escape mutations that maintain good biochemical function. And the actual observed escape mutations shown here in red are among those 4. And in fact, if we look at other antibodies, so we did the same experiment for 4 different antibodies where we could select escape mutations, we see in every case the mutations that actually arise when the virus is grown in the presence of the antibody are those mutations that are about as far in the upper right as it's possible to get. So basically, evolution is, as you might expect, is repeatedly selecting for the mutations that both escape the antibody and maintain what's presumably good viral fitness. What I think is really cool is we can not only explain sort of which mutations tend to arise under antibody pressure, but we can also explain why the virus has a very difficult time escaping certain antibodies. So for instance, antibody CoV2 2165 did not select any escape mutations even in 56 trials. So we tried 56 times to grow the virus in the presence of this antibody, and 0 of those times did an escape mutation arise. So if we go back to our escape map, we can see that there are actually 3 sites where mutations do abrogate binding of the antibody to the RBD. And even when we subset on the mutations that can be reached by a single nucleotide change, there are 4 sort of evolutionarily accessible escape mutations. But again, this is just showing one biochemical phenotype, which is binding of the antibody to the RBD. I'm now going to project the other 2 biochemical phenotypes onto this map. And you can now see that 2 of these mutations that escape antibody binding are actually really bad for the expression of properly folded RBD. So this is D420Y and A475P. So they're probably just not tolerated by the virus for that reason. And then if we look at the ability of the RBD to bind to ACE2, we see that the other 2 escape mutations, N487T and N487K, are really bad for the ability of the RBD to bind to ACE2. So essentially, for this particular antibody, there are no accessible mutations that escape binding that retain good function of the RBD. And that presumably explains why the virus can't escape from this antibody. Now, obviously, there might be multi-mutation epistatic pathways that could arise if there was sort of extended selection. The virus could get many different mutations in the same background, but that's certainly going to be a much slower evolutionary process. And so I think this is really cool that we can actually sort of take these phenotypic maps of how mutations are affecting this protein at the biochemical level and really explain both if and how the virus is actually able to evolve to escape from antibodies. And another use for these data is actually in designing antibody cocktails. So some of you may have heard that many of the companies that are developing therapeutic antibodies to fight SARS-CoV-2, so for instance, Regeneron has an antibody cocktail, and I believe AstraZeneca is making an antibody cocktail. They take two different antibodies and they combine them together because the idea is it's harder for the virus to escape two antibodies together. So typically, traditionally, antibody cocktails have been made by what are called non-competing antibodies. So for instance, CoV2 2499, this antibody, and CoV2 2094 select escape mutations in different regions of the RBD. This one selects them in the receptor binding motif, and this one selects them in the core of the RBD. And those two antibodies are what are called non-competing. So this little matrix in the upper right is showing if you bind 2499, antibody 2499 to the RBD, you can't bind more 2499 because you've already bound it up, but you can still bind 2094. So the antibodies don't compete for binding to the same site on the RBD. And the same thing is true if you bind them in the other order. But there are also antibodies that bind to the same region of the RBD, like 2499 and 2050. They both bind in this receptor binding motif area. And these two antibodies compete for binding to the RBD. So if you bind 2499 first, you can't really bind 2050. So those are competing antibodies. And typically people would not use competing antibodies in cocktails because people worry they might have the same escape mutations. But in our case, we can look and we can see what the escape mutations actually are because we've mapped it out. And we can see that even though 2499 and 2050 compete for binding to the same region, they don't share any escape mutations. And sure enough, when you make either of these cocktails, sort of the traditional non-competing antibody cocktail or this sort of competing antibody cocktail of antibodies with distinct escape mutations, you can see that in neither case can you select any escape mutations, even in many, many replicates, although the virus can escape either antibody individually. So I think this just sort of shows how we can use these nuanced maps to design potentially escape-resistant cocktails of competing antibodies. Okay, so I've now told you how we can build these maps and how we can use them to understand viral evolution under very strong antibody pressure. These maps can also be used just to inform surveillance of ongoing viral evolution when we're not sure what the selection pressures are. So SARS-CoV-2 isolates are being sequenced at a tremendous rate. Here's a plot that's already about a month and a half outdated. And even in sort of early September, there were already 100,000 SARS-CoV-2 sequences that had been deposited in databases. And although this virus emerged essentially from a point source in the human population and initially was highly homogeneous, there are increasing numbers of mutations that are sort of showing up as the virus continues to circulate and evolve. And so it's very valuable to know, could any of those mutations affect the ability of the virus to, let's say, be recognized by monoclonal antibodies that are being developed as antibody therapeutics, or potentially going forward by polyclonal sera that's elicited by vaccination? And so we think a really big use of these phenotypic maps we have is you can just cross-reference the evolutionary information with the phenotypic information. So what this is showing is for each of the 10 different antibodies that we've looked at, we can sort of see what are all of the sites where mutations have an effect on the antibodies binding to the virus, and then we can see, do any of those sites have mutations? And I hope what you can appreciate is actually for every single antibody, so every column here, there's at least one black square. So for every single antibody we've looked at so far, there are at least low-level mutations observed in the circulating viral population that escape binding by this antibody. Importantly, all of these mutations are currently quite rare. So the most abundant one here is 36, and the denominator is nearly 100,000. So they're all very rare right now, but I think this suggests that perhaps with continued viral evolution, mutations with antigenic effects, quite conceivably could become abundant. And so the ability to sort of know ahead of time which mutations will have antigenic effects should be really useful for surveillance of ongoing viral evolution. Okay, so to just sort of conclude, I sort of introduced this concept of phenotypic maps, where what we're really trying to understand how mutations to the SARS coronavirus 2 receptor binding domain affect what we think are the key biochemical phenotypes of this protein, namely its folding, its function, and its antigenicity, or ability to be recognized by antibodies. I then explained to you how building these maps is really useful for understanding viral evolution under antibody pressure, and also how it can be applied to surveil viral mutations that are arising during natural evolution. And going forward, we're really interested in sort of expanding these maps to look at the effects of mutations on, for instance, human immunity elicited by prior infection or vaccination, and also understanding sort of the ability of the virus to evolve to infect different hosts by binding to ACE2s from different species. And so I'd like to end by just thanking the people who did and contributed to this work. So the three people that especially deserve the biggest thanks are first Tyler Starr, who's a really amazing postdoc in my lab, who is the only person in my lab prior to this outbreak doing this sort of yeast display work, and over the span of about 7 or 8 months was able to sort of extend his entire approach to build these biochemical maps I described to you, and is now doing a lot of really cool work to build on that. Ali Greeny is a really excellent MD PhD student in my group, and sort of once Tyler was able to start extending his techniques to SARS coronavirus 2, she decided to put aside her other project, which is on something else, and start working with Tyler. And then together, the two of them have made a really strong team, which has gotten a lot of really cool work done really quickly, I think. And then Sarah Hilton, who's a former graduate student in my group, who built a lot of the interactive visualizations that I was showing you when I was discussing these data. I'd also like to thank our collaborators in the Crow lab at Vanderbilt, who isolated the particular monoclonal antibodies I was discussing today, and were very helpful in lots of aspects of this work. And then I'd like to thank a number of other members of my lab, the Crow lab, and of other collaborating labs, including those of Sean Whalen, Neil King, and David Wiesler. And of course, I'd like to thank my support from the Fred Hutch, the Howard Hughes Medical Institute, Genome Sciences at University of Washington, and the NIH. As I mentioned, the link to the slides I presented is available here. And if you're really interested in sort of looking at mutations in this virus, or engineering the RBD, or understanding things about the RBD, the visualizations I discussed are available at these links. And then there's lots of raw data and computer code here if you really want to dig into any of this. And I've tried to make this talk not overly long, because I know Zoom talks get pretty boring usually. So I'm going to end here, and I'd be happy to answer any questions.
Thank you, Jesse. That was a fantastic talk. And with that, we are ready for questions from the audience. Audience, please raise your hand, or you are welcome to type your question in the chat box, and we'll call you. All right, we have a question from Tim. Tim, go ahead, unmute yourself, and please ask your question.
Hi, Jesse, that was fantastic. What I would like to understand is, given that there's no antibiotic-like resistance pathway to drive these evolutionary or just mutational drift kinds of things, how do you think about why a virus would do what it's doing? Since, you know, I would think it would take large-scale prophylactic antibody treatments in order to drive a mutation in the real world. So can you say something?
That's a great question. So sort of Tim's question is, what's driving these sort of antibody escape mutations that we do observe at low frequency? And I think, as you suggest, the answer right now is probably essentially nothing. So right now, not everybody, but like almost everybody who's getting infected with SARS coronavirus 2 doesn't have any pre-existing immunity, because they've never been infected or vaccinated before. And only a very small minority of people are taking any sort of antibody therapeutic. So I think that's in fact why all of these mutations are at pretty low frequency. I mean, viruses just have high mutation rates, and just due to sort of neutral processes, every 1 or 2 sort of hosts that a virus goes through, it will pick up another mutation, not necessarily in spike, but somewhere in the genome. And spike is a decent, about 10% of the genome. So we'd expect some mutations to sort of come up at low frequency, as I'm presenting here, just due to neutral evolutionary processes such as drift or hitchhiking or things like that. And that's why I think we see quite a few mutations that affect susceptibility to these antibodies, but they're all at very low frequency. So what might happen sort of in the future? So one possibility is, even if there's no immune selection, eventually some of these mutations will drift to high frequency just by neutral processes, but that could take a long, long time to happen. But it's possible that the situation you described right now, where almost the entire population is naive, that's probably not going to be the situation a year or two from now. At that point, probably most of the human population will have some form of immunity, either by vaccination or prior infection. And then there's a possibility that the virus would then start to really undergo antigenic drift, where it's fixing these immune escape mutations at a much higher rate, such as, for instance, human influenza does, because at that point, viruses that have mutations that escape from antibody binding would have an evolutionary advantage and would sort of selectively propagate in the human population. There's no guarantee that will happen. So if we look at other RNA viruses, we know that, for instance, influenza A virus evolves to escape from antibodies and undergoes a rapid rate of antigenic drift, which is why the flu vaccine needs to be updated regularly. But something like measles doesn't undergo appreciable antigenic drift, which is why the measles vaccine has always been effective. And you often hear people talk about how coronaviruses have a lower mutation rate than flu, so they might not undergo much evolution. The rate of evolution will probably be slower, but I don't think that's actually like a very well thought out or nuanced explanation. So although coronaviruses have a lower rate of mutation than flu or measles, they still have a pretty high rate of mutation. I mean, it's about 5-ish fold lower when you consider the larger target size of the spike compared to hemagglutinin. So the per protein mutation rates maybe about 5-fold lower than for flu. And we know that mutation rate isn't everything. So measles and flu have about the same mutation rate, but flu undergoes antigenic drift and measles doesn't. So, the rate of functional evolution isn't just determined by the underlying mutation rate. So I think it's an open question what will happen. And you're certainly right that right now, I think we're just looking at sort of mutations that have arisen stochastically, but there's certainly the potential that there could be a lot of antigenic evolution as population immunity builds up. And I at least interpret our data, which I showed at the beginning of the talk, where a lot of sites in the RBD seem to be pretty tolerant of mutations, to suggest that there is a fair amount of potential for such antigenic evolution. Although we'll really have to check back in a couple of years to be sure.
Thanks.
Hey, Jesse, I've got a question to build on top of that. So have you looked at it backwards? So looking at the mutations that do seem like they are under positive selection, and at this point, pretty much we agree that D614G probably does convey phenotypic advantage. So how does D614G look in your?
Yeah, no, that's a great question. And so probably the only mutation, certainly the only mutation in spike anyway, that there's really ironclad evidence for at this point, or I mean, evidence is never ironclad, but that I would say it seems pretty clear is under has been under positive selection as D614G, and that mutation actually isn't in the RBD. So that mutation is, if you look at the structure of the spike, that mutation is in the S1 domain, but it's sort of lower down in a different region. So we didn't actually measure the effect of that mutation directly, so we can't say anything about what that one's. I mean, I believe the papers that have showed that it has, various benefits to the stability of the spike and probably viral transmissibility and titers, but like our data themselves don't say more about that mutation.
Yeah, I mean, obviously this is wild. Some people say that, 614G isn't really conveying a selective advantage. And if it is,
yeah, I mean, and I think at this point, I mean, obviously this is not my work, this is reading the literature. I mean, I think certainly when 614G sort of initially hit the news, there were a lot of people that were pointing out that although it was going up in frequency, the evidence at that time about whether it conferred an advantage or not wasn't that ironclad. It's hard to prove that. So that was definitely, I mean, that's what people were saying, a number of months ago. I feel like at this point, that situation has changed dramatically. So there's been some really nice work. I think it's like Boltz et al., which is a group in the UK. I know Andrew Rambeau and several other people are part of it. They showed really clearly that the mutation D614G does appear to be conferring a transmissibility advantage on the virus. There was a really nice paper, which I think just came out in Nature like a couple of days ago, that showed that not only these lentiviral pseudotypes, but actually in real SARS coronavirus 2 and both primary human airway cultures and I think Syrian hamsters, the D614G virus had an advantage. So I would say at this point, D614G, to the degree that you can actually prove something, in an experimental model and by looking at natural data, I would say it's at this point pretty clear that D614G is really advantageous. So it wasn't necessarily clear several months ago when it first hit the news.
Right, absolutely. But again, there's more controversy about its mechanism.
Yeah.
But, as far as I can tell, a vague consensus is that it is, an indirect effect on spike binding. So why wouldn't that show up in your assay?
Yeah, no, so it's great. So, I mean, the two things it seems to be doing are probably stabilizing the spikes, or two of the things that might be doing are stabilizing the spikes, so keeping more of the spike in the pre-fusion conformation. And then it might be shifting the balance of the RBDs more to an up conformation. And the reason that doesn't show up in our data, I mean, this is a limitation of our approach, is we're sort of looking at the RBD in isolation, not in the context of the full spike. So we don't see mutations that are sort of affecting, I guess, what you call the quaternary orientation of the RBD relative to the rest of the spike. So that's the reason we don't see that is sort of would be a limitation of our experimental system.
Right. Okay. All right. Now I've got a question about glycosylation. So, but given that yeast glycosylation is similar, but fairly different from human glycosylation patterns, well, why do you think the assay is so predictive of human results?
Yeah, I think it's a great question. So, so yeast do put N-link glycans on things, and there are several N-link glycans in the receptor binding domain. But I think the yeast glycans tend to be more mannose rich. And we haven't, there's been a lot of work on this even preceded SARS coronavirus 2, so in the context of SARS coronavirus 1, that yeast expressed RBD seems to have pretty similar conformations. And the antibodies that we've been looking at, not very many antibodies to this virus. I mean, antibody epitopes, unless you're looking at really weird, heavily shielded viruses like HIV, antibodies tend not to bind to glycosylated epitopes. So the glycans, I think, although they're there, they aren't tending to play a big role in most antibody epitopes or in the interaction of the RBD with receptor itself. It's certainly possible that could be, a shortcoming of our approach, but to the extent that we've identified mutations that we think our assay isn't measuring correctly, they haven't had anything to do with glycosylation. I think the bigger shortcoming of our measurements is there's certain regions of the RBD, particularly near this lipid binding pocket, where I think there's important packing of the RBD with the rest of the spike that we're missing. So at least based on what we've been able to assess so far, when our system is limited, it more has to do with missing certain quaternary interactions between the RBD and the rest of the spike. And we haven't really seen any evidence of things that we think are sort of inaccurate in our assay that have to do with glycosylation, although it is a formal possibility for sure.
Yeah. All right, let me just remind the audience, come on, you guys, surely you have excellent questions. But lacking any other questions, I'm just gonna keep going. Have you thought about using this approach to help address the controversy of how it evolved? There's been a lot of work in the last couple of months. We know that there were circulating bat coronaviruses like RATG13 that are quite similar. There was a big kerfuffle over pangolin MP789. Although there's a recent paper saying, conclusively ruling out the possibility of recombination with the pangolin virus. Again, I don't know how ironclad that is, but have you thought about maybe phenotypically walking back to RATG13 to establish that there would be a clear, viable evolutionary path?
Yeah, no, that's a great question. And Tyler, who's in the upper left of my slide there, who's this really brilliant postdoc leading this, who actually has a lot of background in in sort of protein evolution and ancestral sequence reconstruction from working with Joe Thornton, is looking at exactly these questions. So he's looking at the ability of lots of different sarcovirus RBDs, including some ancestrally reconstructed ones, to bind to different ACE2s, and hopefully it will help shed some light on these questions. I don't think it's necessarily going to provide a definitive answer though, because even when you look at these ancestral reconstructions and build back, you can sort of only, your resolution is limited by what's actually sampled on the phylogenetic tree. And the truth is, like, something like RITG13 is actually like a decent ways away from SARS-CoV-2, and there's nothing in between, you know, so no amount of experiments can tell exactly what's there, but I think they could shed some light. And
Yeah.
other ways that our experiments shed light on this. So as you noted, there's a lot of sort of unusual and unexplained things about the sort of origins of this virus. So in the case of SARS-CoV-1, it was quite clear that it was coming probably from civets into humans, probably from bats to civets. And there was actually clear evidence during the early phases of that epidemic before it was curtailed that there was evidence of mutations that were clearly adaptive, including mutations that enhanced ACE2 affinity that were sort of coming up, as you might expect if a sort of only partially adapted virus hit the human population. In our data, we can see that there are lots of mutations that allow SARS-CoV-2 to bind to the RBD better, but none of them show strong evidence of being selected. So that would suggest that by the time this Wuhan 1, human 1 sequence was isolated, the RBD already sort of bound to ACE2 well enough. And so certainly we don't see any evidence of the same phase of adaptation in SARS-CoV-2 as we did in SARS-CoV-1. So it's possible that either somehow, whenever the virus first hit a human, it had cryptically evolved all the ACE2 affinity it needed, or maybe that sort of acquisition of the ability to bind human ACE2 happened sometime before any of the sequences that have been released. But I think it's a very interesting question, and I think hopefully our data can shed some light on that, but I think further information about that's going to just require identification of more sequences that sort of span this gap from the known animal sarcoviruses to SARS-CoV-2, which is in humans. And I think as long as there remains this big gap with not that much sampling between there, it's going to be really hard to figure out exactly that.
Yeah.
Thank you, Jesse. Okay, there's an audience question here. This is from Nina. Nina, would you like to ask your question? Yeah, go ahead, unmute yourself.
Hi, it was great talk and thanks for sharing all the data. It will be really interesting to look at it. I was really curious, so among all this escape mutation for the antibodies, there are a number of mutations in the protein core. I'm curious whether any of this mutation can trigger the successful RBD expression and potentially like shift the receptor for the protein.
So just to kind of clarify, you're asking if any of those escape mutations in the core are like sort of remodeling the entire conformation of the RBD, is that right?
Yeah, correct.
So we can't say that 100% for sure because we've only, in the data I presented, very little and in ongoing work only partially sort of overlapped our experiments mapping the effects of mutations with structural characterization of these epitopes. But based on what we've seen and inferred from sort of mapping the mutations we've seen on the structure, I think all of the escape mutations we've observed, I think, are directly in the antibody paratope interface and are just abrogating binding of the antibody to the RBD without sort of dramatically remodeling the RBD conformation. And I think that would sort of make sense because the RBD is under strong purifying selection to maintain this conformation in order to bind to ACE2. So I don't think we see any evidence of that. And I think the core mutations that were showing up there in orange in some of those maps, I mean, this is partly my fault for unclear nomenclature, but when I say the RBD core, it doesn't entirely mean inside the core of the protein. It just means the part of the RBD that's not the receptor binding motif. So the structural biologists have, at least in some of the definitions, divided the RBD into sort of the receptor binding motif, which is the part that binds ACE2, and then sort of the rest of it, which is away from ACE2, that they call the core. But some of that core is actually still surface accessible. And so I think all of those core mutations are actually probably in antibody interfaces nonetheless. So that's my interpretation of the data, but we don't have structures to go along with what everything I showed you, so I can't be like 100% sure on that.
Okay, thanks.
Thank you, Nina. Thank you, Jesse. So here's a question. Are any of these antibodies, are you performing translation studies on this? I mean,
Yeah, you mean like using the antibodies in humans or something like that?
or even before that? I mean, some experimental systems like we have mice, hamsters.
Yeah, so we're not, so for instance, our collaborators in the Crow Lab are actively involved in that, and two of the antibodies that they isolated are the two antibodies that are being, for instance, developed by AstraZeneca, and I think are going into phase 3 human trials. In our case, we're not really ourselves trying to advance antibody therapeutics in terms of like having our own antibody molecules. We are now working quite intensively to repeat the type of escape mutant mapping I've shown you for the antibodies that other academics, but especially companies, are putting into phase 3 human trials. And I think that would be really advantageous for a number of reasons. For instance, it could help understand if there are breakthrough viruses that still manage to infect or mutations that are selected in people that are prophylactically treated with these antibodies. Are they actually escaping antibody binding? It could also really be useful to sort of see if any of these antibodies would need to be updated as the virus continues to evolve over time and figure out the best ways to combine the antibodies together. So we're very interested in that area, but we're more coming from the idea that other people are sort of finding and isolating the good antibodies, and our approach that I described here is a really useful way to provide information on how the virus could potentially escape from these antibodies. And yeah, we're definitely trying to translate the approaches I showed you into getting data for the antibodies that are currently, being advanced in humans by companies like Regeneron and Eli Lilly and AstraZeneca and stuff.
Yeah. Hey, have the companies been willing to send you aliquots, or?
It's been sort of in some cases, yes, in some cases, no. I'm like not, and some have been really great to collaborate with, but in all the cases, it's like under various CDA things. So like part of the reason I'm not showing that data is like, we haven't done anything where we won't be able to share the data, but some of it, like we can't share it when it's in progress. We have to wait until everybody's looked at it. So in some cases, yes, in some cases, we've just been able to sometimes find the sequences by digging through patents and things like that and just making the antibody ourselves. So it's been kind of a mixed bag there.
Yeah. Jesse, that's amazing. This was really a heroic study.
Well, thank you. I really appreciate the chance to chat. And if there are any more questions, let me know, or people are free to email me or something if you have questions about any of this.
Yeah, no, thank you for your openness, both in the talk and also with all your data and slides. It's really greatly appreciated.
Thank you, Jesse. Appreciate it.
Thank you. I appreciate it.
Okay. All right. Take care.
Bye.